Yeah, so good evening, everyone. Today we have a concluding meeting of the Bioinformatics Club seminars, and today we have a final discussion with German Demidov. German, hi, can you hear me?
Yep, I can hear you. Can you hear me?
Yes, perfect. Great. Yeah, I think that we can start with, as usual, with the introductory part, and then proceed to the discussion. Today's topic is the genetics of undiagnosed rare disease cases. That's the sort of the bonus meeting, which we didn't plan to hold, but we initially drafted the schedule for this session, but this idea basically emerged as we were discussing the germline variant calling and CNV calling. We thought that it would be nice to have another discussion, which would be specifically designed to talk about undiagnosed rare disease. And yeah, as I've already mentioned, this is the concluding meeting of the spring session, which turned gradually into the summer session. And yeah, this is the concluding part, and I think we've already covered quite a substantial range of topics, and hopefully this will be a good ending to this session. So before we jump into the discussion, I'll say a few introductory words about what we're talking about today. So basically, we have already covered the germline variant analysis, which we've mostly, when we discussed that, but mostly focused on the rare disease cases, the diagnostic pipelines. And we've talked, I think, quite in detail about different aspects of variant calling, the different technical issues which arise, which are related to variant calling in medical genetics. But I think that we have not skipped, but probably have not covered in that much detail as we could. The issue of basically interpreting the results and finding the actual genetic variant which is responsible for the disease in a particular patient. And yeah, we've talked about it a little bit for both short variants and more complex types of variants, but we've not considered all of the specifics, which of course would be impossible in any given seminar, because that's likely something for a multi-day course, and usually people who are interpreting rare disease, they of course have a lot of things to learn, and I think that all of the people who are doing these kinds of interpretation, they learn day by day, basically, with every new case. So there's a lot of subject and a lot of things to consider. But generally, when we are talking about medical genomics, we consider that our ultimate goal in this kind of analysis is the identification of a definite causal genetic variant or pathogenic genetic variant which drives a disease in a particular patient. And the interpretation of this result of the next generation sequencing experiment is usually performed within the framework of the so-called American College of Medical Genetics and Genomics guidelines, or ACMG guidelines, which have been introduced back in 2015, and there has been a lot of attempts to improve them in different ways. But basically, for those who are not that familiar with this ACMG framework, the idea here is that we have 5 basic classes of variants regarding their pathogenicity or possible impact on the disease. We have the pathogenic variants, and this is the most solid class in terms of the causal impact on the phenotype. Then we have the likely pathogenic variants, which do not have that much of a support of their pathogenicity. And then there are variants of uncertain significance, which actually is the group into which the majority of variants identified in any sequencing experiment fall. So basically, this is the most abundant category, the variants for which we don't have enough evidence to classify them as disease-causing or benign. And then actually there are likely benign and benign variants, abbreviated as LB or B, respectively, which actually are not considered as relevant for Mendelian disease. And the ICMG classification framework is built around the so-called criteria or different types of information are being used to classify the variant. And these include all sorts of different information, including the basic type of variant, its localization within the sequence of a gene, its frequency in populations, and different results of computational predictions. And these different criteria, they have different strengths. So there is one criterion, which is the very strong criterion, which basically corresponds to the loss of function of a gene for which the loss of function is basically a known and well-characterized disease mechanism. So this criterion is used and applied to variants which cause premature stop codons in the coding sequence, or frameshift variants, or splice site variants. So these are usually the three which are considered good candidates for this PVS1 criterion, which is the strongest one, and has actually been quite extensively sort of extended by different groups. But actually there have been multiple additional guidelines on how to apply this criterion, because actually there are some cases in which even if the variant appears to be a loss of function variant, it's not actually causing the loss of function of a gene. It usually is very much related to the transcripts, the transcriptional isoforms of the gene has, which are expressed at different levels, or the location of a variant in a particular part of the coding sequence. Usually the part that is closer to the end of the coding sequence, in the close of the 3' end of the gene, due to various factors, including nonsense mediated decay and others. And as I mentioned, yeah, there is one PVS1 criterion, and there's a bunch of other criteria which are strong, and other criteria which are moderate, and then another supporting criteria which are even less sort of valuable. And again, as I've mentioned, there has been a lot of work done with regard to the PVS1 criterion and how to apply it to different variants with different location and different genes. And actually, this is not the only criterion which has been amended in this way. I think there are working groups which try to provide extended guidelines for various criteria. There have been multiple attempts to improve the computational predictions of pathogenicity, and actually try to provide the interpretation of scores which are generated by tools for computational pathogenicity prediction. The different cutoffs for these scores, which may lead to different criteria and different strength of the same criteria to be used in final variant classification. So there's quite a lot of work in this direction. And still, if we're talking about the final outcomes, or the rate at which people receive the final definitive molecular diagnosis from a next generation sequencing data, this rate tends to be lower than we'd love it to be. And this is a figure which is taken from a review from back in 2018. So that's quite an old one, and the figures which are shown here are also relatively old ones. But still, this was the data for 2018. For different experiment types, for different types of approaches to sequencing that can be used. For example, like the trio-based whole genome sequencing, the trio-based whole exome sequencing, or other approaches, including target gene panels analysis. And basically, as you can see from this figure, the diagnosis rate, even from the trio-based whole genome sequencing at that time, was estimated around 40%. And over the years, there have been quite a lot of disagreement with regard to these rates from different laboratories. Some laboratories claim that the rate should be much higher, and they see a much higher rate of up to 60 or 70%. And there were people who have reported even lower diagnosis rates. So there seems to be quite a lot of variability in terms of these numbers. between different laboratories, between different groups, between different centers. And I think that over the recent years, these figures have gone up a little bit, but again, it is my personal impression that they have not gone up dramatically or as much as we would love them to, given that actually the technology has been a standard and a routine method for diagnostics in the last at least 10 years, probably. So that there was there were multiple problems that this identification of the definitive causal variants, and there have been quite a lot of discussions regarding which factors, in fact, drive these decreased diagnosis rates. And we've, in our first couple of meetings in this seminar session, we've talked about the bioinformatic aspects of short variant calling, and we've covered some of the factors which indeed influence the diagnosis rates. But one of the factors, which is also important to talk about, is the fact that just we do not have enough knowledge about the mechanisms of disease. And that's basically what has driven the idea of performing a periodic reanalysis of next-generation sequencing data in the clinical setting. So basically, if we have a patient who did not receive a definitive molecular diagnosis, it's a good practice to reanalyze the same data after a year or maybe several years following the initial attempt. Because due to the accumulation of knowledge with regard to the mechanisms of disease, new genes which are linked into new diseases. New genes are always described and discovered, and the databases containing information about both genes and genetic variants are constantly updated. And this leads to quite a substantial increase in the rate of diagnosis for these undiagnosed cases. Again, this rate is also, the estimates greatly vary between different articles. This is one of the first, I think, systematic reports. They claimed the additional diagnosis for about 10% of cases after a reanalysis attempt. And I think that in many works, in many reports, we can see figures of up to like 20 or 30% of undiagnosed cases receiving a diagnosis after a second attempt or after several years. So this basically illustrates that as we gain more and more information about which genes are responsible for disease and which mechanisms of variant pathogenicity are there, and we improve our ability to predict the pathogenic potential of the variant, we really increase the rate of diagnosis, and the patients actually receive their diagnosis, which in many cases leads to actionable clinical decisions. And I think that there are also a couple of studies which have reported that patients who have received their diagnosis after this reanalysis actually have received not only the diagnosis, but after that certain interventions or therapy which has been altered or prescribed in a way to match the actual genetic cause of the disease, which has introduced clinical benefits to the patients. So this is actually a very important topic. But still, again, even if we consider the reanalysis, we still have quite a lot of cases which still are not diagnosed in the end, and this is what brings us to the topic of today's meeting. So basically, there is quite a substantial fraction of cases which do not receive diagnosis, at least at the first attempt to do so. And then there's the question of basically what can we do with these patients and how can we obtain the final diagnosis. And there have been quite a number of efforts and organizations and consortia established to solve this particular goal. So first there is the Undiagnosed Disease Network project, which is basically aimed at obtaining finding the genetic causes of undiagnosed disease. There is a SolveRD project, which German actually was a part of, if I'm not mistaken, and I hope that he'll share his experience in this particular effort. And but there are other efforts. For example, in Genomics England, have sequenced this 100,000 genomes project in the UK. They've had the so-called rare servoir database or rare servoir framework, which they've used to map new genes for rare disease. And actually, there was a reference which is provided here. So basically, that's sort of an overview of what today's topic is about. So we will dive into more details into how and why these efforts to solve actually undiagnosed cases, how they are conducted, and what are the sort of take-home messages from these initiatives. Yeah, that's it for my introduction. And yeah, so here I think we can start the discussion. Yeah, so German, maybe if you have something to add, maybe to the introductory part, if you think that I missed something important, yeah, please feel free to comment before we proceed.
No, I think we will uncover everything further, but I think it was a very good introduction. I don't see anything that I could readily add. So we can just go through our points and let's see.
Yeah, great. Yeah, that sounds like a plan. Okay, so then I'll start probably the first question, which the first issue which I've introduced during my introduction. So this issue of diagnosis rates. So again, I've mentioned that for several years ago, there have been quite a lot of discussion about what are the ultimate rates from different technologies. There was quite a lot of debate with regard to whole genome versus whole exome sequencing, which they've also participated in a little bit. Yeah, but again, I am aware of figures which people have reported several years back, but I'm not that much aware of the figures which people usually have these days in the leading sort of facilities and centers that do the diagnostics. So if you could say a few words about the actual rates of diagnosis which you see in your center, and maybe elaborate say a few words about this maybe technology debate like whole genome versus whole exome, etc.
Yeah, sure. So regarding the numbers, like how many patients we can diagnose based on the DNA analysis, routine DNA analysis, because yeah, there is advanced analysis, but let's say about the standard of care in different countries is different standard of care. In some countries, still panels. Some are using exomes, and here in our clinic, we recently switched fully to genomes. I don't think there will be any more exomes there. The main determinant of how big is the percentage of solved cases, except the professionalism of the bioinformatics team plus the clinical team, is the disease group you work with. So for eye diseases, for example, the percentage of solved cases is actually very high. For some of the forms of eye disease, is close to 100% actually. So if I'm not mistaken, something like retinoblastoma, some particular diseases, there is more or less one gene responsible for this, and yeah, we will do Sanger, and like 99% of the cases can be resolved. But some disease groups, like the autistic spectrum disorder, this one, even if it's for sure genetic, even we see it in the like family members across generations. Even in this case, the rate of solved cases is around 30%. So the answer is, on average, in our clinic, we would say we will solve like 40%, but it all depends on the composition of disease. So in our clinic, we are specialized in neurodegenerative disorders somehow in Tübingen, and yeah, there it's around 40%, so close to average. So the main determinant still is the disease. I would put it like this. Not even the technologies that we use. Some clinics are specialized in like hematological disease, immune deficiency disease, They will have their own ratio, and some specialized in malformations or autistic disorders, they will have different ratios. So it's not exactly correct to compare them and to say like one clinic resolve 90% and it's much better than the clinic that resolves 40%. Here in this case, we need to take into account the complexity of the underlying case. So the short answer is 40%. The long answer is like it really depends on the composition of the diseases. There.
Yeah,
And
actually, I think it. Sorry.
No, I just forgot to say about the exams and genomes there. It was the second question that you had. And there is an increase in the exams and genomes, but short-read genomes are they're not allowing to resolve many of the structural variants, which are undoubtedly disease-causing. And like running in front of the train, let's say it like this. Even long-read sequencing, even high-depth long-read sequencing, doesn't have 100% specificity, sensitivity in terms of structural event calling. So there is still a lot of work for bioinformaticians, not even for clinicians. So it's directly related to bioinformatics.
Yeah, I think actually, I think that we can elaborate on this and different determining factors a little bit later on. But in general, yeah, what you're saying is actually in perfect concordance with the review which I cited. Yeah, because they've also reported that this 42% is sort of an aggregate figure across all disease groups, and then indeed between the different disease groups, you have quite a lot of differences, yeah, and in variation in terms of the actual rates. And yeah, it's quite nice that there was this concordance with the previous data, which actually I think emphasizes the fact that despite all of the recent improvements, like basically from 2018, we don't still see quite a substantial gain in terms of the definitive diagnosis rate from routine procedures, not mentioning the advanced analysis, of course. Going forward from the actual diagnosis rate question to the further issues. So again, we have 40% of cases which receive on like the aggregate 40% which receive the diagnosis from the routine part, and then we have this additional analysis. So first there is the advanced analysis for the unsolved cases. Which requires quite a lot of specific effort, which we'll talk about a little bit later. But then there is the periodic reanalysis issue, which basically is repeating the same routine procedure, but after a certain time point for the same case. So first, do you have this practice that's like performing the same routine procedure after a while? Or not, and if you do so, what's the increase in the diagnosis which you see in this particular case?
Yeah, indeed, many efforts of many hospitals which do this solve the same problem are now concentrated in periodic reanalysis of their existing patients. So the attempt to solve the cases that were not able to solve before. And in the recent European Society of Human Genetics meeting, there was quite a, let's say, breakthrough talk, and they spoke about reanalysis on the fly. So basically, each time the new variant on ClinVar released or new gene disease association is connection is performed, the reanalysis is done kind of automatically in the background, but it's still a questionable thing. In the reanalysis, the biggest issue is that, like from perspective of Germany, I can say insurance doesn't pay for it. So reanalysis is more or less the initiative of the hospital. So it will be for the case which you are able to solve once again, sorry, not once again, but finally solve, insurance will pay for additional analysis, but they will not pay for the negative cases which you tried to solve, but you weren't able to solve. So all these negative cases are just like you're doing it in your free time. Let's put it like this. So it puts additional barrier there. So we do perform the analysis in-house, but there are different types of reanalysis. So there is reanalysis, for example, bioinformatic type of reanalysis. If you change the version of the tool, like some much better variant caller, it may be structural variant caller, it may be CNV caller, And then you can actually analyze your whole cohort, perform make a big Excel table with all the new variants that you were able to discover in important genes. This is one type of analysis. Another type of analysis is when you involve the clinicians and where the new, for example, once per year, there are maybe, I don't know, 100 new associations between gene and disease. And once per year, you may basically update your gene lists, which you use for filtering, and then you do this type of reanalysis. So in both of these reanalysis, the main issue is bioinformatics. So now the main bottleneck is the time that clinicians have to spend again on looking at the cases they already looked at. And the only way to resolve it is with the use of, I will say, like very the thing that everyone is annoyed of. It's using the artificial intelligence and the machine learning, like everything which actually can help the doctors to reduce significantly the time they need to spend looking at the same genomes, same patients. Another type of reanalysis is when you apply different technology. So, like for example, you try to solve the patient with the exome, you try to solve with genome, and then you could do like long-range genome or RNA-seq. This is the third type of reanalysis and all of them worth to be done, but very accurate cost-benefit analysis has to be done. All of these things cost a lot. And doing this analysis, you have to be sure that you will be able to help some patients. And unfortunately, the economics here is dominating the science. So it has to be cost efficient.
Yeah, so maybe thank you for this explanation. Yeah, but maybe I will return a little bit to the my original question regarding the let's say increases in the how many patients actually receive the diagnosis following the reanalysis step. So you've mentioned that there are several kind of types of reanalysis, which depend on the actual what you do. Basically taking the same data or generating new data, etc. So if you would maybe say a few words, like, do you have any idea, maybe estimate as to what proportions of cases you'll be able to solve basically by just updating the software, by updating the like doing this updated gene list analysis, or maybe employing other technologies and like specific efforts from people.
Yeah, I think I have quite a, let's say, unique experience in this because I worked as a part of the SolveRD project, which involved like 40 different medical genetic centers. So groups which are trying to solve their own patients from their own specific disease group, like group that is specialized in muscular dystrophy, group that's specialized in like eye diseases. And these groups, they use their own bioinformatic pipelines, and they all of them are trying to use this SEMG criteria, but they apply it with different stringency, like this. So despite the attempts to make this SEMG classification to make it uniform, there is actually quite a lot of freedom there. So you may one person may say that this variant is pathogenic, and one person may, looking at the same criteria, still say that this variant is likely pathogenic, for example. So we collected the data from 40 different groups all around Europe, and the first thing that you notice there, it really depends on the quality of bioinformatic pipeline. So many of the medical centers, they don't pay enough attention to strengthen their bioinformatics side. They, for example, they can buy existing solution for genetic analysis from some industry, and they don't invest a lot of R&D there. The groups, sometimes the institute is rather small, so they don't have enough money to keep their bioinformaticians there. And you can see it because you just take the data, you touch the data, and you immediately start to find the pathogenic variants, which were not found by the bioinformaticians before. And it's not because a new gene was discovered, but because, for example, CNV calling or structural variant calling was not properly designed there. And indeed, performance, like this increase of diagnostic value, when you try to reanalyze the same case, really depends on how good the job was done in the first patient. So if your primary the place where the analysis was done the first time did the job really well, it's really difficult to solve any cases out of thousands of unsolved cases. Then you already need to spend a lot of time and you need to have luck in this. And mostly the cases indeed will be because of new gene was discovered. The performance of bioimplementations vary a lot between different institutions, and that was the main driving force, I would say, from my side. I have to say, in this SolveRD project, I have quite a bit weird specialization. I almost didn't touch the point variance. So the point variance with indels and short variance, other people were doing this, and their experience. Most of the clinicians already really quite experienced with interpretation of point variance, like missense variant. From my experience, you talk with anyone, any clinician, even like after like several years of experience, they're already good in this. The issues start when the type of variant become unfamiliar, and it's like some weird variant. So it's both bioinformaticians can like artificially reduce the performance of some particular center, or clinicians who are not experienced with this type of variant here. So the answer is it depends on how well the job was done the first time, and with new omics, let's call it like this. So you have, for example, exome, and then you have money. Within SolveRD, we had money to do additional analysis. Then you can do whole genome after. And if whole genome fails, you can do RNA-seq, for example. Then I would say RNA-seq is a very cool thing. This type of analysis increases by a lot the diagnostic rate. I really believe in the power of RNA-seq where it's possible. It's like we cannot take neurons and do RNA-seq on the live person in neurons. So the answer is everything is nuanced here. And if the job was done not so good in the beginning, you will resolve a lot with the analysis there. The goal of all this project, like SolveRD, is to make the analysis everywhere more or less uniform level, uniformly good. I would say the educational role is very important. I hope I answered the question, but
Yeah, sure.
I have just too many thoughts about this thing, so I feel that sometimes I'm getting quite far away from the original question. But
that's fine. Actually, in the first place, that's why actually I've decided to have this discussion, because I think that your thoughts on this issue are absolutely valuable. So yeah, please don't hesitate to share anything if you'd like. Maybe I will also elaborate on this question a little bit. So you've mentioned the performance of actually we see that there is not only the performance of the software, let's say, which may improve, or the data which may appear, but also the performance of the people, actually, and the experience of the people who are doing the analysis. And I think that several years back, I think that at least in some cases, I've heard from some people that, for example, the actual results of diagnostics may also quite substantially depend on the clinician, not in terms of interpretation of the data, but in terms of the initial description of the phenotype. So basically, the phenotype is poorly described, and then you are stuck with trying to find the specific genes which you'll use for filtering, because indeed, for example, if you're looking for a neurodevelopmental disorder, you'll use probably a different list of genes compared to a metabolic disorder, etc. And then the description of the phenotype is really it really matters. Again, at least that was something which I've heard from some people several years ago. But I have to say that this was said also in a setting where people, for example, didn't have the whole genome data, but they were actually working with some types of data which were either target gene panels or clinical axons, which were basically where probably the technology matters more in terms of that. If you select the gene panel to sequence, and if this decision is made wrongly, then you basically just don't have the data to find the pathogenic variant. And I'm not sure that the same problem with the phenotype description is basically the same, has the same level of importance when you have data such as whole genome and whole exome. Though I believe that it still does. So yeah, the accuracy of phenotype description. So maybe if you can say a few words about do you like really observe any issues with these with the quality of phenotype description, and not necessarily in SolveRD, but just basically in your routine practice maybe as well. Does it matter that much in your experience?
Yeah, definitely. Phenotypic description importance a lot. It's like now the fashionable term is deep phenotyping of patients. There's a standard for this. I think it's called tip to toe standard. So more or less this everything moves towards standardization nowadays in this field, but we are still not there. And it depends a lot on the clinicians who perform the original checkup for on the patient. Personally, I work mainly with so-called HPO terms. These are like it's a tree of phenotypic traits. There are like parent terms, there are child terms. Like more common goes to more specific on the bottom, and more common are on the top. So when the clinician is in doubt, they basically take terms more close to the beginning of the tree, and when they are more sure, they take the leaves of the tree and they put it in the phenotypic description. And as bioinformatician, you can of course work with this data as with the basically you compare distance on the trees. So you take the genes, and we know that gene cause some disease, and disease cause some phenotype there, and we compare the description of the patients with the description of disease, and then with some machine learning we can kind of automatically compare phenotypes. It helps a lot in the interpretation, and if the phenotype is written poorly, we cannot, of course, use these instruments, and we use wrong genes for filtering, and we have a lot of chances to miss the pathogenic variant. It's not that easy to estimate how many of the cases were not resolved because of the bad phenotyping, because we will never know this. It's quite expensive to ask the patient to come once again, and another doctor has to check this patient and to fill out his phenotype. Another doctor has to perform it, and it's not that frequently done. What I can say, again, in this SolveRD initiative, there are several research groups in Tübingen submitted their own samples, and we didn't know, but patients go not only to one neurologist, they go to 2 different neurologists. And these 2 neurologists submit the same patient, being unaware that the another group also work with this patient, because patients struggle to find the diagnosis, and they don't trust the doctor, and they go to another doctor, and the another doctor also doesn't find the diagnosis, and they submit quite different phenotypes there. If the neurologist in one group is specialized in, I don't know, some anatomy of the brain cells, they write the phenotype in one way, and the other neurologist will write in the other way. But to be honest, most of these same patients submitted by different clinicians, if the phenotypes are quite similar. But for example, I was in the undiagnosed hackathon in Nijmegen. It was attempt to solve unsolved cases, but in the format of hackathon. So more than 100 people came to one scientific center for 2 days, and we were trying to solve collectively. People with different specializations, mathematicians, clinicians, professors of biology, so every type of specialist. And it's actually these multidisciplinary teams are required to solve these unsolved cases, because this task become too complex for the narrow specialist. The expertise should be super broad and super good there. We were solving one case, we were trying to solve it, and we found very good variant in the gene which causes primary phenotype is cataract. Instead of cataract, it causes a lot of malformations of the face, autistic features. Everything is in patient except the cataracts. There is no cataracts there. And so we cannot diagnose the patient because the phenotype seems to be non-matching. The main thing which should be in this patient, because this variant is supposedly pathogenic, is absent. So we had to contact the original clinicians, and they checked the patient card, and they found out that the eyes were examined by the junior doctors, who most probably didn't notice any cataracts or nothing. And the patient is also young. So this cataract appeared in the young age, but the patient is much younger than this. So this patient would be undiagnosed if we if we wouldn't contact the clinicians. And the reason why they also didn't diagnose this was, first, bioinformatics there was not perfect in that clinic. It's in some Asian country. I forgot, from Thailand, I guess, some of them. And the second is the phenotype was done by the junior doctors. I cannot say they're bad doctors, they're just junior doctors, and maybe they didn't have enough experience this, so they didn't report any eye abnormalities. This is example how the not exactly, it's actually correct phenotyping. It's just like people did what they know, what they knew, but it can indeed stop prevent from diagnosis. Yep.
Yeah, that's it's a very nice story. Yeah. Actually, regarding the phenotyping, one short follow up before moving on. So I think that quite in several years they're having quite a lot of effort to do sort of deep learning on, for example, face recognition. Yeah, where you have the phenotype of the patient, like the picture, which is converted to basically like these HPO terms using some kind of model. So do you have any experience with these kinds of models? If you do they work? Like, do they really help you or not? Just short.
In short terms, short answer, in the routine clinical practice, we were trying to introduce this deep learning things which help. You take a picture of the patient, and then the artificial intelligence suggests the syndromes, which syndrome the patient has. And you may not find the genetic variant there, but for example, you may perfectly know that the patient has this syndrome even without the genetic variant explaining this. It helps in this case, but in our experience, our doctors kind of refused to use this software because they said they can do it themselves. It's more or less, I would say, probably a question of professional pride. So they are very useful, in my opinion, but it's not very easy to convince everyone to use these deep learning things. So it's still not implemented at the level of a clinic, I would say. Gestalt Matcher and the second one, Phase 2 Gene, I think there are two types of software. And speaking about the artificial intelligence in general, there is a lot of effort. As I said, the main issue now is to reduce the human time that is spent on each case. So there is a lot of efforts, a lot of startups, and a lot of big companies who are working in this field, trying to automatize this process of genetic diagnostics, trying to help to make suggestions to the clinician, so they don't have to check 100 variants, but check like 5 of them. And there's a lot of competition, and even the people who work in these companies, in these startups, they all know that only a couple of these hundreds of companies will remain there. Currently, I struggle to say which platform will be used in 5 years. And yeah, a lot of competition, a lot of progress there. In this hackathon in Nijmegen, it was a unique event also because the partners from the industry were invited, and they were all showing us their tools in action. So trying to solve the cases using the artificial intelligence tools. Yeah, I have to say a couple of them were really useful. Sometimes the results are not relevant. I was very skeptical myself, but then I touched the platform, touched the data, and we solved one case with this artificial intelligence suggestion that this particular variant has to be causal. We made investigation in depth, and yeah, the platform was. I don't work for Illumina, but the platform was from Illumina. Other platforms were also quite cool there, but 2 days is not enough to work with all of them. So I worked with like 3 or 4, and it's fascinating. Gestalt Matcher, the tool developed in Bonn, where you upload the picture of the patient and it suggests the syndromes. It was also used, and it was also very useful. Yeah. So these efforts will definitely the future of the field.
Yeah, that's thank you for the answer. Maybe Alan, just moving on to the actual or not moving, but continuing with the discussion of what drives or what improves the diagnosis for the undiagnosed patients. I maybe would like to turn to the issue of what factors or what manipulations may be helpful in order to get a better diagnosis. So basically, yeah, there are, I think, quite several categories of things which may be done. So you basically can have the same data, but analyzed with a different approach or with a different tool, looking for other types of variants. For example, like not only the short variants, but the structural variants, or specifically looking for something like repeat expansions. There might be, if you have the same data and the same type of variant, but looking for additional evidence for a particular variant. And then there is, of course, the option to just generate other types of data, which you already mentioned, just using different technology like long-read sequencing or RNA-seq, for example, as you've mentioned. So maybe to start with, maybe you can say a few words regarding these three options. So basically, if you have globally the two options to use the same data and try to dig into that, or use a different technology, which one basically is expected to give like a better result? So whether in which of the two you should invest, and if we're talking about reanalyzing existing data, so basically, is there any relevance to like looking for things which have not been looked previously, or on a relative scale, maybe how many patients will get the diagnosis from just like reinterpreting the same kind of variants versus trying to get new variants from the data?
I would say reinterpreting of the old variants is unfruitful. People who actually did this, actual genomic interpreters who did it in, again, this SolveRD project, they said that they went through thousands of variants and they solved like several patients, because most of the time you already seen these variants and you already tried to interpret them. It's very rarely that the circumstances change and the variant was reported in like CleanVar or something like this, or the variant was reported in the same amino acid that is pathogenic, for example. If it's reported in same amino acid, the missense variant, then it also increases by a bit the pathogenicity there. So checking the same variants, most probably you will lose your time. Unless these are new genes. If they're new genes, then you're not actually checking the same variants. You're looking for the new variants in the old data. If you didn't perform copy number variance analysis before in your exome data, and you just start performing, and sometimes it happens. Sometimes I get emails that people just start doing CV analysis despite doing genetic diagnostics for like 5 or 10 years. Or it's crazy, but sometimes it happens. You may increase like 3-5% of the diagnostic value. It may lead to substantial increase. The trick that everyone is doing now, it was fashionable like 3 years ago. You have a large cohort of your patients, unsolved patients. You look for the large, I mean several thousands. You scan for mobile element insertions. It's super rare cause of rare disease, but if you scan several thousands of patients, you'll find one causal variant. So small increase, but still. The issue with the new wet lab using the new technologies is the money. So most of the insurance companies, they don't pay for the reanalysis with the new technologies, absolutely for sure. Yep. So if you have money and you have exome data, then it totally makes sense to do whole genome. But in each individual case, it will be very different. Sometimes for some diseases, it's much better to do trio, for example. So you have one patient, and you know that the variant is likely de novo, that it appeared only in the patient. Then you better sequence the mother and father. I don't know, can we call it generation of new data? Yeah, I think it's generation of new data, but it's not even from the patient, it's from the parents. Very individual. For different disease groups, the choice of the next analysis in the let's say pyramid of diagnostics, diagnostic pipeline, it's individual. I would say it's very nicely described in the gene reviews. It's a web resource on gene reviews. You can look for the disease, and then they describe how many cases are solved with the microarrays, how many cases are solved with Sanger sequencing, and then NGS, etc., etc. Like this. So individual. The answer is if you have money, do all the analysis.
It's nice. Actually, so I think you've mentioned, previously mentioned the RNA-seq kind of analysis, and this is for me a little bit. I'm not that familiar with using of RNA-seq for Mendelian disease genetics. Yeah, I perfectly understand what, for example, the long reads bring into the market with the better sensitivity for structural variance and ability to actually map complex variation. But in terms of RNA-seq, again, I've never worked with RNA-seq in the rare disease setting. So what actually is the way, how do you apply RNA-seq data, and what does it tell you, which you don't see from basic genetics? So is it like monolithic expression or something?
There are different types of consequence which you can see on the RNA level, and it doesn't always work. For example, the gene may be not expressed in blood, and you may simply not, for many neurodegenerative diseases, you need to take biopsies of the cerebellum or like somewhere from the brain, and it's not done, as far as I know, only post-mortem probably, and it has to be done immediately because of RNA-seq. It brings a lot to the table, and recently there was a big progress in bioinformatics side of this thing, because when you perform RNA-seq analysis of the rare disease patients, you don't perform classical differential expression analysis group versus group. You perform the analysis of like everything against your one particular patient. Your patient, since the disease is rare, then the molecular consequence should be rare, and there should be like one unique change in expression, like reduced expression, increased expression. So it requires special methods, and I think the method was developed in München in TUM. It's called Drop Pipeline, and I think many people use it currently. It was published in Nature Protocols or something like this. So it's in a very good journal, and indeed, it's a very useful instrument. So what people are looking for is the alternative splicing, of course. They have sophisticated methods to measure that this particular splicing was not seen in the cohort of controls there. They look for increase or decrease expression, as I said. You can look for the whole gene expression level, and also they look for something else, which I actually don't remember. But indeed, this monolithic expression, you see in the decrease or increase expression. So there are several other type of consequences, but the causes which cause these particular changes, it may be non-coding variants, for example. So you might see an RNA level some consequence, but you see nothing in DNA, and then you go deeper into DNA level, and then you see some interesting non-coding variant. If it's de novo, again, if it's in mother and father, absent, but in the kid is present, you may try to validate this finding after. Many structural variants, again, they can be seen in RNA level even without the DNA level. For example, you do exome sequencing, and inversion happens. And when inversion happens, it doesn't change the copy number, but it may break the gene in the middle. And you see the decreased expression, but on exome level, you will see absolutely nothing. And on genome level, we all know that our reference suffers a lot. This human genome 38, it's not T2T, and even if it's T2T, alignment of short reads to T2T will not help. So if the breakpoints of structural variants are somewhere in the badly mappable regions, we will see nothing. And in the RNA-seq level, we will still see this. So we kind of we solve the task without finding the genetic variant. It happens often. The final goal is indeed to find the genetic variant, of course. So it sounds quite weak. You still can write diagnostic report based on the RNA-seq only, but people will normally try to find the underlying gene. So it brings a lot to the table, which is very difficult to find in just DNA analysis by itself.
So in terms of writing the diagnostic report, so basically, if you see the differential expression for a particular gene, which is uniquely observed in the patient, so basically then you'll do the conclusion based on the gene, so the association of this gene with the disease, without finding the variant, just stating that okay, this gene is associated with this disease, and that's likely means that we have this one.
For the discovery of new genes, now the standards are super high. So you cannot actually discover a gene having only one patient. Unfortunately, now to write a paper in a good journal that you discovered a novel gene, you need to spend much more efforts looking for the patients with the same disease, which is this initiative of exchanging patient information between different countries, different continents. These are international gene matcher, I think, these things. So you're looking for patients with very similar symptoms or the variants in the same gene, something like this. So to discover exactly new gene, RNA-seq is not enough. But for the diagnostic report, yes, if you see the decrease of expression and you don't see the genetic variant, but the known mechanism of disease is loss of function, I think you can write the diagnostic report. But you still have to specify that genetic variant was not found, but still it may have already therapeutical implications.
Yeah. Yeah, thank you. Maybe also quite a few minutes ago, you've mentioned in particular the role of non-coding variants, which may impact the expression, which you will be able to see on RNA-seq. And maybe here I would like to ask you. So I think over the years there have been also quite a lot of let's say discussions regarding the proportional contribution of non-coding variants to rare disease genetics. Yeah, and here I will maybe briefly cite, and maybe I can not only cite but also briefly share the picture. So basically, yeah, we've done in the last year, we've done an analysis of the proportion of coding versus non-coding variants in the ClinVar as it changes through time. So basically, here we have this picture again showing just from a piece of from our paper. So the thing which we wanted to ask is basically whether we will see an increase in the proportion or a decrease in the proportion of non-coding variants among the ones that are reported as pathogenic, and basically the proportion of coding variants remains constant over time. So that's a good question whether or not we should interpret this as the fact that there is a limited contribution of non-coding variants to the genetics of rare disease, but at least we can state that with the current technologies, we still see that more than 90% of all pathogenic variants remain coding, and this proportion does not virtually change over time, at least in the last 5 years. So basically, the question, my question is, so what is the maybe a proportion or the number of cases in which you have seen a diagnosis which can be performed using a non-coding variant? So actually, is that common when you see this non-coding variation and you can somehow prove that it's pathogenic, or you didn't have this at all? And if yes, if you've seen these kinds of cases, maybe you can speculate a little bit as to whether or not we'll be able to better predict the consequences of non-coding variants and actually bring them to the table the same way as coding variants are now in the near future.
I've seen very few non-coding variants which were classified as pathogenic and not reported in ClinVar before. So most of the time, if it's reported a non-coding variant, and if it's reported in the diagnostic report, most of the time it's because it was discredited. That's kind of universal things. So to go into non-coding variants, the issue is like we can interpret the role of the variant in the gene, like the missense variants or loss of function variants, some frameshift variants. But for non-coding, we obviously cannot do this. Currently, the software, the Splice AI, it helps a lot. So I see a lot of progress in the reporting of the variants because of Splice AI scores. So if it's de novo and the Splice AI score is really bad, then this variant probably worth to be functionally validated, for example, or RNA-seq can be done on this patient. But due to these large cohorts of whole genome sequences that appear right now from the rare disease patients, there are a lot of cool discoveries in non-coding genome, and I think everyone who works in this field, everyone knew this story, which was published like half a year ago, like 0.5% of all neurodevelopmental disorders were actually resolved by some non-coding RNA, which is you cannot interpret the variance there as like missense because it's not coding thing. But if you have a lot of data, you may do statistics on huge cohorts. The problem is like when you have genes as unit, you may kind of merge the variants which are in the same gene, different variants. But in non-coding, you cannot exactly merge them. So if they are in the same region, people are trying to merge them if they are like they're using like 5 kb windows or something like this, and they try to merge the variants which are like there, but so far with not so much success. So I would say we need to have a recurrent non-coding variant to be able to resolve it with statistical method. Yeah, the answer is we will see. I see we massively. I think we massively underdiagnosed the non-coding variants, and I hope there will be huge progress. But it has to be done in multi-omics and whole genome approaches. Currently, there is the first good background database which appeared is this Genomic England whole genome there, which is kind of let's say available. It's not exactly available for all the researchers because it's super regulated. The access to this special IT system that they do, but is the first steps to this direction. I think the progress will speed up in this area.
And so, as far as I understand, so you think that so what maybe if you can guess, or maybe you would not like to guess, but still, regarding to what you expect can be the proportion of. So for now, let's say we have 40% of cases which we solve routinely. We have 60% that we don't diagnose. So maybe if you have sort of an estimate of what proportion of these would you expect to be from non-coding variation, or again, this was a pure speculation. I understand that there is no maybe basis to like really confident, let's say, or hypothesize the objectively this proportion. Maybe you have some sort of expectation as to what proportion of the cases could be solved with better understanding of non-coding variants.
From pure intuition, no, without having any substantial evidence, I would say like 40% solved with coding, maybe 50% in the end with improving of the methods, and 20% we will solve with non-coding. So less than in coding, but still much more than we solve right now, because now it's quite a dark matter in the diagnostics.
Yeah. That actually I would be very concurrent with my impression. Yeah, so my intuition will be also that the contribution for non-coding variation should be a little bit less than coding, but probably much more than we can see now, because now we have like around 10% for known pathogenic variation in there. So maybe again returning back to this issue of different factors that may influence the diagnosis of this undiagnosed problem. If we're talking about other technologies, we've touched a little bit upon the RNA usage of RNA-seq, but maybe we can then talk about the long reads, and I think you've already mentioned that the long reads allow you to have much better accuracy of structural variant analysis. But maybe I can also, my hypothesis is that there are some regions of the genome which are not accessible to short reads, even for short variant mapping. Because the region is absolutely repetitive. The canonical case here is RSMN genes, SMN1, SMN2, the spinal muscular atrophy genes, which are not resolvable virtually without long reads. With short reads, I think there are several methods which try to do some sort of complex probabilistic inference of which variants are in which of the paralogs, but this is very specific statistics. And here I anticipated probably if you have long reads, you can also look for short variants located within the poorly accessible regions of the genome. So maybe if you can share your experience with regard to long reads. So have you seen cases where long reads can provide not the causal structural variant, but to actually show some short variant which could not be seen using the short reads? Have you seen any of these?
Yes, definitely. I think it was even published in the preprint. I'm co-author, but I was not exactly working with the short variants again. But it was done. I think in shorter genomes there was kind of a gap there. So like the coverage was really dramatically reduced. It was still mappable, but for unknown reasons to me. There was almost no reads. It happens. It happens quite often through the genome. So they couldn't see the SNP there, and it was known gene, pathogenic variant. Very easy story. If you do, the sequencing was more than 30x short read, but still the variant was not discovered. But it's indeed super rare, I would say. I don't know, 0.1% maybe something like this. It would be so. It is a problem, but mostly for, I would say, structure event and repeat expansion. For them, the long reads are extremely beneficial, and for short variants, I would say still they produce more false positives, the long reads, than actually generate true positives. So there is pro and contra, and currently with current level of nanopore and PacBio technology, I would say with the standard level of coverage, it would be more contra than pro in terms of short variant.
Okay.
I'm unaware about any validated diagnostic solution based on long reads currently because of the short variant. No.
And yeah, so this is, I think, would be a good place for me to ask you about because I know that you have quite recently published the very nice EJAG paper with the structural variant analysis in the undiagnosed cases. So maybe if you can share some insights which you can share from this particular effort, maybe things which you'd like to highlight from this particular study, with structural variant analysis in these undiagnosed cohorts.
Yeah, I'd be glad to do this. I think I'm quite happy how it worked out, the whole story. Indeed, there is a paper published in the European Journal of Human Genetics. We looked for structural variants in exomes. So this is quite a weird thing to do because, yeah, normally you need whole genome. Most of the time, the breakpoint of structural variants are in between the exomes. But it was my personal initiative to run a structural variant analysis in exomes because I knew it's going to work. Since it's not in standard clinical practice, many people were against this. They were saying we're wasting our time and resources, but they still did the analysis. And I think like around, I think 8 out of 6,000 unsolved patients were solved exclusively due to this analysis. We wouldn't be able to discover it with alternative methods. It was just, we solved more patients, but this particular 8, I'm pretty sure that they would remain undiagnosed otherwise. So this is a classic story. If you have initiative, if you're really eager to solve patients, you can make some extra steps and try to diagnose patients even in the situations where everyone was saying it's a bad idea. It's in some sense it was a bad idea because I annoyed clinicians asking them to look for many variants which were not pathogenic in the end. But in the end, it ended up in a nice paper. A lot of manual work. There are like 20 authors in the paper, but I think it should be like 60 authors. Yes, there should be much more authors because many people put their efforts there. Solving undiagnosed patients will require extra steps, I would say. There is no easy steps now, and you will often fail. Some of the projects which I start, like for example, looking for mosaic copy number changes in exomes, they were not fruitful almost. There are several variants, and I'm not strongly confident in if it solved the case or not. But this 1 out of 10 that I started actually worked out. So it's just a lot of work if you like the topic, if you're eager to solve the cases, if you like to solve these riddles, then it's probably for you. But yeah, it's a very I think it's a very nice story, and not in terms of science, but in terms of the collaboration, in terms of how the process can be done there.
yeah, I'm just showing this paper for those who are interested, maybe to see the title, etc. Yeah, so that's basically what I was mentioning. Yeah, thank you for sharing this idea. Yeah, and maybe here this is also a good point for me to ask you. So given all of the things which we've discussed regarding the different factors and different options, maybe sort of an outlook and there's a prospects for the future research and practical efforts. So if we imagine some sort of an ideal world in which you have different technologies, etc., what would you consider to be a sort of a good or the best possible pipeline in terms of like methods and what to look for in a rare disease case to maximize sort of the diagnostic yield? Again, if you can like think of a world in which we don't have also like financial limitations, etc.
One of the initiatives I take part in, this undiagnosed hackathon, they work under the ideas that we need for each patient, we need to do the maximum, and they do all the analysis for most of the patients, like short-read whole genome, short-read exome. Most probably, short-read exome usually is remains from the first attempt to solve the case. So it's not a resequenced exome, but exome which is kind of legacy exome. Long-read genome, long-read RNA-seq, normal RNA-seq, methylation profile, metabolomics for many of them. Metabolomics is super useful in many of the diseases, more than genetics maybe, because it makes strong suggestions of these specific diseases. So you may not know the genetic variant, but you may know that the patient has exactly this disease. So it's very in-depth approach there. And I would say it's unfeasible to analyze all this data at once unless something will help to humans. Currently, we don't have enough genetic interpreters, smart genetic interpreters who are experienced with all this type of data and know what to do. And probably you need to spend like 5 hours per patient to just examine the candidate variants there. Here, I would expect the biggest breakthrough from bioinformaticians, not even bioinformaticians, it's maybe outdated term. People who work in machine learning and artificial intelligence field, I think they need to push here. So the dirty job should be done by the computer. Currently, we put all of gene filters, allele frequency filters, all these classical filters, but it's still the medical doctors still spend too much time evaluating the candidates. It has to be done, especially in the reanalysis context. Primary analysis is not that bad, like you can spend several hours, but if you want to reanalyze the patient in 1 year, you know that you're not going to be paid for this, then the artificial intelligence is the only way to go. I use this term, but it can be any statistical learning solution. I just use this term artificial intelligence because the data structures are really complex. And I think the best is to use these complex methods. The problem is the diseases are rare and training deep learning models require a lot of training samples. So this is kind of an issue, but hopefully with more coverage of patients, more countries who will be paying for the genetic analysis from the insurance. Still not all, even European countries pay for the genome. Some countries don't pay even for exome. Yeah, I'm not sure how it is in other countries, but yeah, in Germany it was also stepwise process. And with more patients sequenced, with more collaborative efforts, these are not just words. I actually felt it myself when I was trying to solve patients. That you have a doctor whom you know from England, you have a doctor whom you know from Congo. They have databases of the patients, and you may ask them, "Did you see any variants in this particular gene?" And once out of 50 times, they answered, "Yes, we've seen it," and it may lead up to a paper and resolving diagnostics of the patient. So international collaboration, it's a real driver, and the artificial intelligence solutions, machine learning solutions, which will reduce the space of the variance for the humans. That will be the main drivers in diagnostics of unsolved cases in the future. Not even the technology, I would say.
That's a nice idea. Well, maybe as a conclusion, yeah, I started sort of other speculative question. Yeah, so we can think of this that even if we have sort of this ideal picture and we have all sort of methods available on all people and artificial intelligence model, etc., we still can expect that there will be a certain proportion of cases we will not be able to diagnose just because there is no actual genetic cause of the disease. So there is quite some fraction probably of the patients for which the reason is not the actual like truly pathogenic variant, but some sort of other issues like certain environmental exposure during development or some interactions between genetics and environment, which will not be able to trace from single sample. And so maybe if you can, say a few words again about whether you think that this is a common phenomenon, and if it is, so what proportion of cases, in your opinion, again purely on intuition. I understand that you probably don't have the objective sort of data to use to make this prediction, but just from pure intuition, what's your expectation of like the ultimate proportion of patients who will still not receive the diagnosis, even if we have like all possible data and all possible methods for their processing?
Yeah, it's a good question. The standard numbers are like 20% of rare diseases have non-genetic origin. Everyone repeats from presentation to presentation. I never traced the origin, but I still kind of trust it. Maybe 20% is a good estimation there. And in my own practice, it happened only a couple of times that after different tests of DNA, they were found out, for example, that the mother were exposed to some chemical during, as you said, the developmental process of the kid, and it made the impact on the kid. It was, instead of genetic, the diagnosis was made based on the exposure. It was not very often. Again, it will require like these causes. I would say they're really truly sneaky. These causes will be for genetic causes we have the evidence, DNA, RNA, something, and for some unknown exposures which happen environment we cannot exactly trace. So I would say this 20% of the is a good number. The 20% of rare diseases are not genetic origin. The issue is like, will the doctors understand? Will they be able to classify the patients who come to them as the genetic origin or not genetic origin? So it's the question to the first stage, I would say, of this analysis is the maybe even family doctor, so not even the specialist, who will say that this is probably non-genetic. Yeah, and it's more clinical question. Tough to say. Maybe like several percent of the cases that come to us to the genetic testing and we try to find genetic solution, they are not actually of genetic origin, but it's extremely difficult to prove. Most of the time, they just remain unsolved for unknown reason, like this.
Yeah, thank you. That's all for my questions. If you have anything to add or maybe say something as a concluding remark, then feel free to share anything if you have any thoughts.
Yeah, thanks everyone for coming. I know it's summer, it's late, and everyone's maybe like walking. Yeah, well, more people probably watch this thing. It's a very cool field. I'm passionate about this field. I don't regret that I came there, and it won't bring a lot of money, I would say, because the diseases are rare, and the farm companies, they are mainly interested in big markets, and the rare disease is rarely big market because it's really rare. But it's kind of rewarding because, when you, especially when at some stage you start to actually see the patients and you know whom you help, and from the human point of view, it becomes really rewarding. I would say that currently there's a need for strong bioinformaticians, and mainly not in the classical way bioinformaticians, but more like in, as I said, specialist in this machine learning things. And I hope that someone who was listening will probably try try if they can help to this area. And, I'm ready to answer the questions on this way. I'm ready to help with all the questions. Yeah, I hope that more people will come, and together we can make a difference. Yeah, that's my conclusion.
Yeah, thank you so much again for coming and for doing this bonus meeting. Again, we didn't plan it. We actually, it was an idea which came us to us on the fly, and yeah, but I think it was still very interesting and valuable to hear from your experience. Yeah, thank you, and thanks to all who have been listening.
Thank you, Yura.
Yeah, this I think we can conclude this series of seminars, and yeah, we'll think of future sessions in the upcoming months and years probably to come. Yeah, thank you. See you all.
Ciao.